The RNA world hypothesis is about life on Earth before the emergence of coded proteins and DNA. So the simple view of it is that there were communities of RNA molecules in some compartment, perhaps inside lipid vesicles. And this allowed two levels of selection, intra-community and inter-community. And there were two catalytic roles for each RNA molecule, a selfish role and a communal role. So as an unfolded template, it acted as a selfish autocatalyst. So it catalyzed its own sequence reproduction. And it competed for communal resources, whatever replicase, ribonucleotide precursors, competed with the other RNAs of the community. Then as a folded ribozyme, it served as a communal catalyst. So adopting some sequence-dependent folding, it cooperated with other ribozymes for the survival and reproduction of the community. Chemists and biologists gradually recognized four different kinds of kinetic behavior. So starting with stoichiometric reactants, these were either substrates that are consumed quantitatively in the course of a reaction or products formed quantitatively. So an example of that today is the conversion of mononucleotides to a polynucleotide product. A difficult idea that emerged in the early 19th century was the idea of a catalytic reactant. An example I have here is how a message RNA directs the synthesis of the cognate protein. So a catalyst accelerates a reaction or equivalently selects one path and product over alternatives. There's no net consumption of the catalyst. It's either regenerated immediately from the transition complex or more generally regenerated through some multi-step cycle. So the mRNA is there at the end of protein translation to do it again. So it qualifies as a simple catalytic reactant. Then a notion of autocatalysis, and there's really two flavors of this. So the first one, convergent autocatalytic reactants, and an example is the role of glycoaldehyde in the Formose reaction cycle. So the reactant is regenerated, and a duplicate of it is generated. It's convergent because the duplicate is either an exact duplicate of the original or something very close with a limited range of variation. So there's a net production of catalyst in an autocatalytic reaction. Divergent autocatalytic reactions, and this is RNA templating a copy of itself. So the original is regenerated, the template remains, and a duplicate is created. The duplicate may be exact, but has the potential to be different, a little different. So there's the ability to form a product that's never been seen before, possibly with activities that hitherto have been unknown. So branching evolution, divergence from that. So again, there's net production of the catalyst, and if it's an autocatalytic reactant. So one of the great papers in the history of biology I don't quite put it up with Mendel's paper, but it's pretty close, is this second paper by Watson and Crick on the significance of the newly determined structure of DNA, genetical implications. And they noted that given one strand of DNA, we'll call it the template, that nucleotide monomers, they didn't know exactly what these monomers would be, but could hydrogen bond with the template chain, just as one polymer chain hydrogen bonds with the other in the finished double-stranded molecule. Anyways, they could pair, and the thought was that if they were correctly paired by the Watson-Crick rules, everything would be sterically right for possible polymerization. So the free monomers could become a polymer templated off a complete strand. So the specific template was needed for this polymerization. They weren't sure whether it was sufficient or whether there needed to be a general purpose polymerase assisting this. And they also noted that this kind of copying doesn't recreate immediately the initial strand, but creates a reverse complement, and that you have to copy the copy to get back to the original template chain. So this is the foundations of molecular biology as we know it. This slide anticipates a lot of the talk. On the left column, we have times in the American billion years ago. So we have a 1 billion year window from the formation of the planet, the Earth-Moon system, to this time 3.6 billion years ago, when there's good evidence that there was cellular life. And the simplest assumption is that it was already DNA life with the steps of the central dogma that DNA replicates itself, transcribed to RNA, and translated to proteins. So the sequence of steps in between are in question, more so, I guess, the timing of those. But you can see it's a lot of things happening quickly. So a prebiotic period in which polymers, random polymers, RNA, peptids, maybe some other things. Then a period of life, early RNA life, this having crossed the Darwinian threshold, in which RNAs could template their own copying. But it was non-enzymatic. They didn't have a general polymerase helping them. They did this by the natural tendency of a new strand to kind of crystallize on the template strand. And peptids, to the extent they existed, were still non-coded. Then a late RNA life, and where RNA copying is assisted by a communal ribosome, the ribosome polymerase. And then protein life, where two things have to happen. The ability to translate mRNA into proteins, so coded protein synthesis. And then the emergence of some useful proteins, including a polymerase, that can then take over the function of the ribosome polymerase. Finally, DNA life, where DNA becomes the double-stranded DNA, the long-term, stable, repairable genetic store. and we have to make RNA from it by transcription, and the proteins by translation of the message. So there's been a lot of prebiotic chemistry and laboratory studies on the possibility of non-enzymatic copying. In the background, primordial flows of energy and matter, the non-equilibrium processes that allow this chemistry to become biology. And this slide just shows very rough grouping. I mean, the energy inputs can be from the sun, an important either direct radiation from the sun of different forms, or secondary, indirect, but still driven ultimately by the solar flux. And then finally, the energy sources can be intrinsic to the Earth, terrestrial. So the volcanism and other forms of magnetism, radioactive decay, various geochemical, geothermal gradients, and then one that's the tidal cycle, which is the Earth-Moon system. And then finally, extensive extraterrestrial impacts bringing matter and energy into the system. So those things are kind of macro looking. A lot of attention to more micro questions of what places and conditions could you concentrate reactants or products. And the opposite of concentration is the diffusion or the exchange. So a lot of questions of compartmentalization and transport. Physical compartments that are considered droplets, aqueous droplets in the air, aerosols, essentially droplets, but in ice, the interstitial brines, kind of droplets in the pores of rocks. Organic compartments that have a carbon-based component to their barrier or exchange. So water droplets in oil, emulsions, micelles, which are kind of the opposite, or closer to oil droplets in water. True vesicles or vesicles, membranous vesicles with selective pores. And then finally, once it's a little bit biotic, kinetic compartments. So either ways of telling yourself that your RNA that belongs to this community is some genome tag, or a way to destroy non-self RNAs, so nucleases. So for the first ribozymes to evolve by non-enzymatic copying, there had to be some templated process. So either something not so processive, ligation, or something more processive, primer extension. And a problem that the copy has to be separated somehow from the template or displaced if there's going to be further rounds of replication. Have to have the nucleotide substrates, so prebiotic synthesis of ribose nucleobases, a way of putting them together. And then the thought is that random oligomers, and adequate variety and concentration, suitable length. So short, non-templated oligomers, and some activation of their leaving groups and appropriate chemistry of the ends so that you can have whatever the ligation or extensions. Another point in this, besides all the goodies that are needed, is something about the stability of the RNA, the conditions that make it stable or allow it to have activity. So I think there's no escaping that this has to be seen as a slow-paced life, this non-enzymatic RNA copying life. And slow-paced isn't such a bad idea, that today the biological arms race in all aspects, behavior, metabolism, makes speed essential. And here, a time when there's very few biotic nucleases and you're not in such competition, it's possible that we just turn the clock speed down. So the backbone and the nucleobases have to be stable. This, of course, is favored by low temperature, so people have proposed eutectic phases in ice. pH, alkaline pH is destabilizing for the RNA backbone, so neutral or acidic. Divalent metals, magnesium ions, can cause strand scissions, or catalyze it, so maybe not too much of that. Intention for folding of RNA and their catalytic activities, it seems usually you need a fair concentration of magnesium and maybe a lower of sodium. So the other thing about all of this in the background is the kinetic pathways and the thermodynamics. So I have traps for the kinetic and sinks for the thermodynamic. So damaged ends, they can't be joined, or a kinetic trap, at least until the evolution of a repair mechanism. Misfolded aggregates or a kinetic trap, they, in general, non-covalent misfolded aggregates with RNAs can be unfolded and refolded, but it takes time, so it's a kinetic problem. There's potential thermodynamic sinks. Long, stable duplex RNAs just take the molecules out of commission unless they can be recognized and chewed up. And then there's things that are, a kludge, kinetic and energetic. So chemists like to call these things TARs, and they're too complicated to analyze and go forward on. Okay. So here's a recent paper saying some of this prebiotic problem might not be as terrible as feared. So the problem was that any prebiotic synthesis that made D-ribose would make deoxyribose and other sugars. And anything that could make the modern nucleobases would make other things. And the backbone linkage is not only the 3 to 5, but 2 to 5, and more than one phosphate in between. So in this paper, they, in the lab, showed that many kinds of mixtures of true RNA components and side products, that the simple act of annealing together for templated non-enzymatic polymerization and then melting apart or dissociating some way and doing it again and again, multiple rounds, tended like a purification by crystallization to produce more or less pure RNA molecules with the right backbone. Another view of all of this period of the non-enzymatic copying is that there are a lot of RNAs, a lot of oligos, and they overlap one another in both senses, and at low temperature they can anneal, giving the possibility of extending one or the other or ligating together. A lot of kinetic sinks are possible, so there's a lot of issues there. But the idea is that stepping back from it, we can imagine that there's one grand canonical circular sequence, not represented by any real molecule, but covered adequately by RNAs in either direction, so that we can replicate the virtual circle by these kind of more ad hoc copyings of their fragments. And the paper points out the genome doesn't have to be one virtual circle. It could be a number of them each. So another important idea was that whatever the repertory of catalysis that folded RNAs were able to do, that could be augmented. And a lot of the modern coenzyme cofactors, in fact, look like they were derived, or they are, biosynthetically derived from nucleotides. So this shows acetyl-CoA, NADH, and FAD. And so the idea is that, well, maybe these cofactors are older than proteins, and they worked with ribozymes originally, and they gave the RNA world some of the catalytic repertory that we associate today with the protein world. One last point on this. Funny title, it's the consolidation of Fuca. So everyone talks of Luca, the last universal common ancestor of cellular life. This is the first. And it's titled Exploiting the Chemical Affordances. So it's different useful ribozymes emerged here and there in different RNA communities. What's needed is some mechanism for gathering together as many of these good things as possible into something that's going to become more like a cellular ancestor. So not one or two activities, but paths and collections. So there's something that vertical gene transmission with the clonal selection is the notion of, each membrane enclosed community or something having an evolutionary future of its own, competing with others, but not exchanging genes. Clearly there's extensive horizontal gene transmission, so there's something akin to the modern pangenome concept for bacterial species, or mechanisms of hybridization that we see more recently, like the endosymbiosis, or plants formed as hybrids. So what could the ribozymes do well early? Evidence from the lab with artificial ones show that could likely make and break glycosidic bonds between the ribose and the nucleobase, maybe act as kinases and cap RNAs, and certainly the fundamentals of ligation and polymerization. On top of this, the most immediate pressing things might have been to improve the metabolism of the compartment of the membrane and its pores, and maybe to help synthesize amino acids and random peptides if they're part of this. So this, I guess this is a review article in Science in '69 by Arthur Kornberg, describing what they'd learned about the DNA polymerase. And so at this point, they knew quite a bit that Watson and Crick didn't know. So first, this would work with apparently any unique template. So it wasn't restricted to some motif or something. It required a primer, and the addition units were the deoxynucleotide triphosphates. It was processive, remarkably processive, a step size of 1 nucleotide. So I would read a nucleotide by adding the complement. So it read from 3 to 5, the template, and it built the new strand 5 to 3. And this is his figure, which I guess he shows the primer and the template. And here is a thymine triphosphate coming in, pairing with the A, the one being added. So an influential paper at almost that time by Manfred Eigen emphasized 3 things: the generality, the fidelity, and the speed of replication. So the generality of replication is really that idea needed for the divergent autocatalyst. So this universality, sometimes people use that word. So it needed to be able to faithfully replicate a template regardless of the nucleobase composition, not requiring any really strong sequence motifs, and despite any sequence-dependent folding. The fidelity, he assumed life evolving under purifying selection, and the idea was that a sustainable genome under those conditions, the length had to be less than the reciprocal of the error rate. So for a genome of an effective size of 1,000 nucleotides, so 1,000 positions that matter, you have to have an error rate of less than 1 misincorporation per 1,000. As far as the speed goes, it's really just the polymerization rate has to be greater than the decomposition rate. So this became the model, the protein DNA polymerase, and Eigen's ideas for finding an RNA templated RNA polymerase for the RNA world, something that would add a single nucleotide processively. So a template, a primer, some nucleotides as substrates. The polymerase would work. It would have a rate. Faster would be better. It would have a frequency of misincorporations. Lower would be better. And ideally, it would put in the right linkage, and then there were problems with the overall reaction cycle too. So identifying the template, especially if there are other RNAs about that don't belong, distinguishing the 2 strands, the plus and minus, if they needed to be, how to make the primer or a anneal one pre-made, where to start and end replication, how to unfold the template and refold it, and then to separate the copy and fold it. So all of these were needed to have a iterative replication cycle. So a lot of engineering and then artificial selection experiments, so starting either with a natural ribosome or working from scratch. This is the secondary structure of the most famous nucleotide polymerase ribosome. One of the things that emerged in this hunt is that a little broader net might be needed. So although modeling on Arthur Kornberg's work, the idea of adding processively one nucleotide at a time, polymerases that added more or weren't so futzy about how many. So here's a paper with triplet polymerase artificial ribosome. So it's 3 bases, but it's a triphosphate, a 5' triphosphate, I believe. And it's not wondrously processive. So it's a little bit more like templated ligation, and but so this is where that has gotten to. So this is the point of the talk. This is an old paper, old in the standard of here. It's 1991 by John Campbell. The title is An RNA Replosome as the Ancestor of the Ribosome. So at that moment, the realization that the peptidyl transfer center and the decoding centers of the ribosome were really RNAs, catalytic RNAs, and the thought that, well, maybe the problem of kickstarting RNA life, getting it beyond that non-enzymatic copying, maybe that used these best-known ancient RNAs. So he published a very short, here's what little I learned about John Campbell online. Caltech undergrad, PhD in immunology at Harvard, did a couple postdocs, and then spent his career on the faculty at the UCLA School of Medicine. So this slide kind of shows the problem as he framed it. There'd be a prebiotic period of making random RNA oligos, early RNA life within non-enzymatic copying, late RNA life with duplication, with this mechanism that we'll talk about. Then early protein life, where RNA is made, duplicated by machinery that's very similar to the machinery that proteins are translated with. And then late protein life, where the translation continues, but a protein polymerase is taken over RNA replication, and the duplosome is retired. And then finally the DNA life. So here's the thought. So the RNA templated RNA polymerase reads from 3 to 5 and builds from 5 to 3. But protein translation on the ribosome reads from 5 to 3 in codons of 3, and then adds an amino acid for each. So he thought, well, maybe before protein translation, there was an ancestor of the ribosome which read in steps of 3, read from 5 to 3 in steps of 3, but rather than adding amino acid, added a 3 nucleotide piece, a duplicon, and built up the duplicate RNA. This great idea, but it came with a twist, a bizarre twist. So that's why I'm calling this twist duplication. So I'll show that. So here's the cartoon of protein translation as he understood it. What's important here, we have the growing polypeptide chain. We have the amino acid on the A site tRNA. That's going to pick up the nascent chain, making a nucleophilic attack there. The codon, the current codon paired with the anticodon. And then we move on. The ribosome motors along the mRNA, or you can think of it, it extrudes the mRNA in the other direction. So for twist duplication, here's the growing copy. Here's the anticodon. Here's the duplicon, which is the Watson-Crick partner to that. And it's at the 3' end of the ancestral tRNA, which dubbed here a donor RNA, a dRNA. So it's going to grow like this. The problem is if we put the original sequence there and then we put the twist copy that's been prepared, it's not a reverse complement. It's not anything so easy to describe. You can stare at it and see what you have to do, the origin of the word twist. But he's completely right. A twist copy of a twist copy is back to the original. So a twist copy, he noted, doesn't anneal to the template. So the problem of having a long, stable duplex unlined isn't there. Still got the problem that the twist copy is a minus strand. You have to do it again to get to the original plus. So there's the twist duplication. You're reading from 5 to 3. Just a couple years after his paper, but apparently unaware and independent, something I've dubbed here true duplication was proposed. And so now we're going to read from 5 to 3 and codon steps, and then we're going to build from 3 to 5 in duplicons. And if we compare this to this triplet polymerase artificial ribosome, the most striking difference is, again, that we've reoriented the template strand. Okay. So this was Weiss and Cherry's idea. There's the template strand on the top, the codon in focus. This is the true copy, not a twist copy, that's been built from 3 to 5. Where at this 5' hydroxyl on it. It's going to make an attack on the phosphodiester bond here, 3' of the anticodon. And so this whole thing here is meant to be an oligo, maybe a little bit like a tRNA. You know, they're not that committal. Gordon, a couple years later, totally commits to the idea that this is a tRNA and all of its features and draws it as such. But they just are saying, well, there's some machinery here for pairing a codon and anticodon and supporting this this nucleophile attacking. And then we translocate this little reading head and have a hydrolytic attack by water. So we've already cut off this piece, and then we're going to cut off that piece. And so what we've done now through this cycle is we've extended the growing chain by 3 nucleotides at the 5' end. And now we've loaded up the next little pseudo tRNA for addition. So the true duplication, you go from the original, you make the true copy, which is just your Watson-Crick looking thing, and a true copy of a true copy is back to the original. So the good news is elongation is a little bit driven. There's a net hydrolysis of one phosphodiester bond, so you have to make one to extend this, but you break 2. The downside, there's now a long duplex unwind, so unlike Campbell's twist duplicate, which wasn't strongly paired, and again, there's a minus strand, so you got to do it again if you want to get the original. So this jumps to 2012. This is the paper I actually saw in teaching a lecture, undergraduate genetics, human genetics on the RNA world. It's just looking for something to beef up the lecture. So I think I saw it a decade ago when it came out. And before I talk about what Noller proposed, I want to talk about what had happened. Those early papers, Campbell 91, Weiss and Cherry 93, Gordon 95, were before the atomic structure of the ribosome was determined by X-ray crystallography, and before some of the dynamical intermediates had been characterized by cryo-EM. So here's a review article, 2009, what the recent ribosome structures have revealed about the mechanism of translation. And this is just showing you the cartoons from the original papers. This was the fertile imagination of what might be going on inside. So here's a depiction of the crystal structure. I'm not going to go through it, but it's here. One of the big breakthroughs was the discovery that in the decoding center, there are certain adenines which interrogated the pairing of the codon and the anticodon in the minor groove, and they allowed a greater fidelity in this pairing than the 2 strands that the tRNA and message interaction alone would support. So this is one of the secrets. It was a 3-strand, 1 strand being the small subunit ribosomal RNA at this critical juncture. So here's Noller's figure. I think this is a little aside to jazz up a talk, this paper, but he produced this cute model. These are his drawings. So there's the template, and these are tRNA-like things with the decoding interaction that are reading. So these are a P site and an A site tRNA, if you'd like. These guys are homodimerized so they're coupled to one of them themselves. And at this other end, the anticodon is building the copy, the product molecule, by selecting a trinucleotide pieces to add in and some unknown ligation mechanism. So we're reading the template and we're ligating the product over here. So this emboldened me because I thought, well, if an expert on the ribosome can propose such a far-out model of something that happened before the protein world, that there was something there to think about, at least for giving a student lecture. And the article had a lot of things I didn't know anything about, like A minor interactions and all of this. But it had some things that seemed obvious and seemed worrisome. So I've dubbed this what he called indirect duplication. And the word indirect is just because it requires a decoding and a recoding, if you like, 2 steps there. So good news is there's no minus strand to copy again. And there's no duplex online. And minor groove interactions can improve the fidelity. The bad news, and this seems to completely vitiate it if I understand it, it's got a degenerate third base. And an ability to copy RNA that gets 2 out of 3 right is simply not what RNA life needs. They need, so those eigenerror rate reciprocals, you need something a lot better. So that's not going to work. A second thing that seems very problematic is the idea that there was going to be these independent matching somehow codes the anticodon and this tetrameric palindromic tail that would pick out the correct dRNA to make the dimer. So keeping a system like that up and running and not drifting. And then the fact that it required 2 decoders, one to read and one to add. The probability of problems seems to go as not a little p being squared and becoming smaller, but an enormous problem becoming exponentially worse. So anyways, that didn't seem likely, but there were some attractive things about it, just as there were some attractive things about the Campbell paper. And interestingly, Noller didn't mention the Campbell paper. Virtually no one did. And if it were seen, I think, and Campbell didn't write about it again and again, I think that bizarre twist duplicate that in his heart, Campbell thought, well, that's pretty strange. And other people who saw it, they thought that was strange too. So I'm going to talk about the best of both worlds, direct duplication. So protein translation, we read from 5 to 3. Noller's indirect duplication, we read from 5 to 3. Direct duplication, we're going to read from 5 to 3. And the one immediate change is note that adding 2 nucleotides at a time, not 3. Some other changes, but so here's the other thing is Campbell parked the duplicon at the 3' end of the tRNA, where the modern amino acid is charged. That gave him the nightmare twist. Parking the duplicon at the 5' end of the tRNA solves that. So here's the copy being made from 5 to 3, just as we're reading from 5 to 3. The duplicon, which is now going to pick up the nascent chain by nucleophilic attack there. And then we've moved on. So, and I've labeled this doublet decoding because we're decoding 2 bases at a time in the template. We're going to get to something called triplet decoding later, so that's why I emphasize that there. So this is the 16 dRNAs you would need for this. So the body of the dRNA is here, the anticodon, it ends up there, and the duplicon which is the Watson-Crick complement of the anticodon at the beginning of it. So the good news was there's no minus strand to copy again, no long duplexed unwind, minor groove interactions available for fidelity. These are the rest of the talk, the second half of the talk. I'm going to go over 6 points about it, some questions, some pretty immediate questions, and some possible answers or bemusements. So first, let's talk about phosphoryl transfer. So this is what we want to do. Here's the P site, the RNA with the nascent RNA chain. Here's the A site with the little duplicon here. And then here are the products. So now we've transferred the chain to the A site, the RNA, and released, not released, but we've freed the P site one. So we want to get from here to here, and the how of it. So one way to conceptualize this is to have the nucleophile adding before the other group leaves. So we've got the spectator, the leaving group, and the nucleophile all there at once. Phosphorane or a trigonal bipyramidal sp3d kind of intermediate. So that can be compared to the formal reaction and the peptidal transfer. So we have the nascent chain, the immunoacyl tRNA, and then the post-transfer. And at some point in the past, people thought, well, maybe this goes through some sort of tetrahedral intermediate. So more and more was learned about the actual peptidal transferase center. And the first thing that's pointed out seems to be an entropic trap for the nucleophile. So it's really mainly constraining and orienting the inline reaction. A second thing that people originally thought it would go through a zwitterionic tetrahedral intermediate. I still thought it goes through a tetrahedral intermediate, but it may in fact only be anionic or such. The second point that was found is how to deal with the protons that move around the charge. Match the electronic movements. And one of the breakout papers was the discovery that if you replaced on the final ribose the 2'-OH, replaced it, made it deoxyribose, or put a fluoro group there, so it didn't have an oxygen with a proton to toss about, the catalytic rate dropped tremendously. So the idea was that this is substrate assisted, the tRNA is the substrate proton shuttle, and that this proton that's originally here on the 2' hydroxyl ends up being the proton on the 3' through a chain of intermediates of 3 protons. It's been modified more recently, and people have made the shuttle longer and added 1 or 2 waters in it. Okay, so just to say, formally we run a phosphoryl transfer. Today in the ribosome we have a peptidal transfer. There's something known of the mechanism. was going to point out that there's another model rather than the modern peptidal transfer center. There's another model to think about. It comes from the natural nucleolytic ribosomes available today. So these are reversible reactions, so cleavage and ligation. So here's RNA. Chemists can worry about the intermediates, but we get the products. We get a 2,3' cyclophosphate and a free 5' hydroxyl. And the reaction can go back. So for the phosphoryl transfer, to make it a sequential cleavage and ligation, here at the P site, the nascent chain, it's cleaved, giving off the free dRNA, and here we have held in the PTC somehow, the nascent chain ending now in a 2', 3' cyclic phosphate. And now from the A site, it's 5' hydroxyl, not this one, but this one, even if it's just luck of the draw. Ligates, and now we have the duplicon extended chain. So all of this leaves open how in the world do the dRNAs get loaded? So here's a reminder. This is the structure by Holly, the proposed cloverleaf structure, secondary structure from the sequencing of the primary sequence of a tRNA. And so just to walk through it, so at the 5' end, there's a phosphate. We go down the leading part of the acceptor stem, and we come through the stem and loop that are called the D stem and loop. We start down the anticodon stem and loop. Here's the anticodon, 34, 35, 36. We come back through the T stem and loop. And we end up here with this unpaired extension, these last 4 nucleotides, including this one they like to call the discriminator, 73, and the universal CCA at the end, and ending in a 3' hydroxyl. And in fact, the aminoacyl tRNA synthetase will charge either on that 3' hydroxyl or on the 2' on that same ribose. So here's how the tRNAs are loaded by the aminoacyl tRNA synthetase. 2 steps. The first one is to activate the amino acid. So here's an amino acid in blue, now on an AMP handle with the release of pyrophosphate. And then the charging reaction proper, so that aminoacyl AMP plus the uncharged tRNA puts the amino acid, and here they show it on the 2' hydroxyl. So how would the dRNA be charged, be loaded? The top shows translation. So there's the tRNA sequence with the standard 1 to 76 residues, the amino acid at the end. It was discovered that the recognition features by which the aminoacyl tRNA synthetase recognizes this is the right tRNA for me, as well as knowing what amino acid, were found in different places, not simply at the anticodon, not simply in the acceptor stem. And then when it's used, after it's loaded, the anticodon is key for reading the message, and then the acceptor stem for donating. So this had created a bit of a befuddle, understanding how the two important features, the anticodon that gives specificity to the tRNA, and the acceptor stem with the amino acid gives, adapts that specificity, gives the right product. How things 70 angstroms apart, at least in evolutionary terms, kept coordinated. So Campbell suggested that the dRNAs, and that's the structure he proposed, with the last three being the duplicon, self-charged, but he self-loaded. He didn't say how. He didn't, it was just one line there. So somehow it loads the duplicon, and then inside the duplisome, you read the template and you donate the duplicon. More recently, a year or so ago, a particular model was proposed based, for Campbell's twist duplication. So the main idea is if this is a stiff L-shaped tRNA or dRNA, two of them might come together in a homodimeric pairing, so that the anticodon of the one could template the duplicon loading of the other. So that was the scheme. I've dubbed it cross-loading because it takes two equivalent dRNAs to load one another. So it's a busy table, and no worries. It's only this last row that matters, and I'm going to go through this, how we suggest the dRNA might be loaded in direct duplication. So this is a cartoon of what we think a dRNA might look like, and it's just literally the 5' half of the modern tRNA we've cut off at the anticodon, and labeled this the open conformation. And the idea is that it exists in two conformations, open and closed. Open is the conformation, the accommodated A-site dRNA that's here in the decoding center and here in the phosphoryl transfer center. The closed is the conformation needed for charging, so that the anticodon can direct the assembly of a new duplicon. And so this, taken literally, then, has an extended stem of 15 pairs, a loop of 8, so there's a total of 38 nucleotides. It's worth giving you numbers and talking literally and not qualifying all of this with, well, it's 4 billion years ago and differences. So let's just walk it through, but I just want to say, what do I believe? I don't know. I'm giving the talk. I'm not the person who has to evaluate it. So that's on you. Okay, so two conformations, open and closed. And here I plugged in actual sequence. How do we get actual sequence? Well, this, I think, is just an alanine tRNA from E. coli. And I ripped off the 5' end. So this is the genuine D stem and loop. And this is extending it to make the closed molecule, and in green, the suggested addition of the duplicon. Well, what do we know about tRNA maturation today? So a lot of tRNAs have an intron at this canonical position between 37 and 38. So if they do, that has to be spliced out. There's a protein splicing endonuclease and different ways of ligating. Then the 5' end is trimmed by RNase P, leaving a 5' phosphate. The 3' end is trimmed, and then the rest of that unpaired extension is added by a nucleotidal transferase that adds the C, another C, and an A. So it does it stepwise, but just one enzyme, and some distant relative of poly-A polymerase, which puts poly-A tails on RNAs. Okay. Well, what about RNase P, the thing that manipulates or matures the 5' end? So it's an endonuclease. It's found in all kingdoms of cellular life. And one of the great breakouts was that it's an RNP, but it's the RNA that's catalytically active. And then it has, depending on the kingdom, one or more accessory proteins. Okay, so it's a ribosome. And what can you say about its substrates and the reaction that catalyzes? Well, just stepping back, looking at it as generically as we can, substrate it likes is an unpaired leader sequence followed by a paired trailer. So that's an unpaired leader, and there's a paired trailer. The examples are simple stem and loop for the paired trailer, so that's a signal recognition particle pre-RNA. The pre-tRNA, it's a coaxial stack and loop. But the products then are the leader ending in the free 3' hydroxyl, and then the 5' phosphate with the trailer. So the question is, could that ancestor of RNase A, it's the catalytic RNA, we'll call it ribosome P, emphasize it was a ribosome, but mnemonic there, could it have been a reversible ligase and nuclease involved in dRNA loading? So let's look on the bottom. So we have a cognate oligo. That means some oligo that ends in the right two bases to charge the dRNA we have here. And here's the free dRNA for that. So the idea is if it could pair and ligate, so that -1 to +1 position, the charging, and now it's extended the base pairing on that stem a little bit. Running the other way, rather than trimming at the bond that it's just made, rather than recleaving when it's ligated, it would perhaps prefer the trim where the unpaired leader begins between -3 and -2. So that's the whole idea. Charges at -1, trims at -3, rather than going back at -1. If it were a non-cognate, so it did not have the stabilization of Watson-Crick pairing there, the trim reaction might tend to simply be the reverse of the charge. So straining this for what it's worth, the possibility is we have something like this. These are the closed but unloaded dRNAs, and these are the feedstock of oligos. This is not meant to show transition states, it's just meant to show some reaction intermediates. So in the paired case, there's some toe-holdish thing, there's nothing going on there. The ligation, and it's, I said not a transition state, but intermediate, but these would be higher energy intermediate, if only because we went from 2 species to 1, so the entropic reason. And then the difference between the incorrect pairing and the correct is that 2 Watson-Crick's stabilization. And then we come down with the trim reaction, where we trim the correct one where we like it. We've trimmed the incorrect one. So now this is a mischarged dRNA, but it lacks that pairing and potentially can be trimmed again, creating 3 products and having rejected the mispairings. So that formally is a kinetic proofreading, so that the fidelity of the loading reaction takes advantage of that extra irreversible step. So now we have a real problem. We've suggested when we load with ribosome P, we have a 5' phosphate, like the modern mature end of the tRNA. But we've suggested that for phosphoryl transfer, we need a 5' hydroxyl. So the end of the trim reaction, there's a phosphate, but what we need at the A site needs to be a hydroxyl. When a free dRNA is dumped, it's got a hydroxyl. To charge it, we need a phosphate. So the suggestion here is there's some ribosome unknown to us which acted as a 5' phosphoryl transferase, swapping the phosphates back and forth on the ends of these dRNAs, and that was needed to couple the loading and the elongation cycles. I didn't like this. It still upsets me. What I thought glibly might work is equally problematic. Concerted transesterification. So we just leave the phosphate there at the 5'. What we want, these would be our starting materials. These would be our final products. There's no problem. It's no thermodynamic problem. It's isoorganic. But it's hysterically, I think, ridiculous. Without building any models with actual bond lengths and angles, I think it's just not possible to have these phosphates near each other for that kind of concerted reaction. But it's maybe imaginable that there's some sort of sequential transesterification. So we've managed to keep our phosphate and go from the substrates to the products as we want. So I leave that to a serious chemist, not a sociobiologist like myself. So I want to talk about the processivity of elongation. So here's the cartoon of the feedstock of oligos being made by mechanisms not specified. The catalyst, the template, the duplisome, the free dRNAs, which in fact are regenerated through the cycle. So all of these things are regenerated. They're all acting catalytically, the ribosome P maybe. So substrates, and then the products are the oligos that are shortened back a bit, the duplicate that's been built up, and one of the curiosities is that the final dRNA is still stuck there at the 3' end. So that idea, what drives that? Well, concentration of the oligos, the feedstock. If we pull out the whole reloading issue, and we just start with the loaded dRNAs and end up with the free ones, this is what we see for the equilibrium. If we make it simple and we just say, well, a reaction, rather than being a lot of steps, we'll just go through one transition state. It's really the ratio of the free to the loaded dRNAs weighted by the stoichiometry. That means how many times that particular dinucleotide sequence is used in constructing the duplicate. So on the one hand, this is a very flat energy landscape. The number of species, the number of RNA molecules has not changed. The number of phosphodiester bonds has not changed. So neither the bonds or the number of ends. We've shortened these ones, the dRNAs, to lengthen this one, the duplicate. If there's some base pairing, like the stabilization of the anticodon and the duplicon, we're losing that. If we're creating some base pairing, some stabilization by folding the duplicate, we're gaining that. But all in all, it looks like this is reversible, controlled by mass action. And so if you really take that literally, then you say, well, can it run the other way? So to make it run the other way, we would want to have some duplicates to chew down and some free dRNAs. And this would be a template-guided exonuclease, I suppose, for chewing down the duplicate and creating a pool of loaded dRNAs. So it's a little reminiscent of the silencing machineries, the RNA-guided machineries today that are used to use a guide RNA to hunt down cognate RNAs. The thing that's hardest to think about is, when running it backwards, is where are the smarts? When you run it forward, the smarts come in right away, the base pairing of the anticodon and the codon. Here, it's like, well, it's only when you're about to leave the A site is now a charged dRNA being expelled. But the trick is the willingness of that charged dRNA to leave is going to depend on the proper toehold pairing of its anticodon and the codon of the template. I won't ramble on that, but anyways, so feedstocks, kind of a nearly reversible thing, didn't like that, so wanted an energy source to give a direction to it in irreversibility. So here's a review on bacterial RNA thermometers. So it's discovered that translation of some mRNAs in bacteria is thermally regulated by a change in the folding of the 5' untranslated region, the thermometer on the message. And these have complicated structures, but they all have critical hairpins. I've just shown the critical hairpins. And the idea is that in the cold, in the closed or folded conformation, the shine Dagarna box is masked in the hairpin, not accessible. That's the start codon, the AUG. So these are two different examples of real RNA thermometers. And then in the warming, the hairpin unzips from the free end, and it may, and real ones typically go through intermediate states. So it's maybe not simply a two-state annealed and melted. So here's a comparison of the RNA thermometers and our idea of a closed dRNA. They're very similar. I mean, of course, I've rigged it a little bit, but stem and loop, some degree of pairing. The RNA thermometers we think of as for gene regulation, and what we want to think about is mechanical work. Can we get useful work from a thermal cycle here? And roughly put, the dRNA is harder to open when it's cold outside than when it's warm. And that's the whole idea. Forget the stuff on the left unless you want to see it. And this graph, so we have the idea we have two temperature baths, one near 0 centigrade and one the human body temperature, some spread of that range. And we want to look at the delta G of opening. So we are here in the warm and we open. According to the RNA fold program, it costs us about 14 kcals per mole, depending on, what we concoct the actual sequence to be. And we keep it open, but we cool it off, and then we allow it to close. And here I've trimmed it by the duplicon. We get a lot of free energy. So we get net useful work here, in this example of about 9 kcals per mole by opening it in the hot bath and closing it in the cold bath. And so our machinery, or the duplisome, is going to be helping ensure that. And what are the hot and cold baths? Well, it's the sun. So this is a 2-cylinder A and B site, if you want, or better, I mean, the two dRNAs, thermal motor. And so the loaded dRNA enters the A site on the warming day. It enters, accommodates. Then in the cold part of the day, the translocation, the movement, or whatever reversibility it has, this clamping or closing of the dRNA, which now can do it because it's given up the nascent RNA that was holding it in the PTC. So the transfers here, enters, accommodates, transfers. Now this one can close, drive the process directionally. And it happens another day, another night, and we're ejecting the one that entered here. So it actually takes 2 days for the one that came in to leave. We thought of this, it seemed so slow, so painfully slow, but very robust. If night follows day, very robust. So the idea is warming drives decoding. It enters, it gets a toehold, opens, accommodates the transfer, which is isoergonic, and then cooling, so it drives translocation, or, it's the ratchet. So translocation, it closes and it leaves. So is that fast enough? And I say slow and steady wins the race. Well, first idea is life was much slower paced back then. So if you grow very slowly, very slow elongation, that's okay if things are stable. So the substrates, products, catalytic intermediates are correspondingly stable. The rate, 1 duplicon extension per day, and let's just give us 6 months of the year as the growing season. So about 360 nucleotides could be added in a year. So if we need to make an RNA as big as today's ribosomal RNA, the large subunit, it's going to take us 8 years. Could be a little shorter. One way for it to be shorter is that the duplosome RNAs are made in fragments, like some contemporary ribosomes, like in Euglena. So more and shorter pieces. Another way for the generation time, the effective generation time to be shorter, is if more than one duplosome can jump on a template at a time. So like the modern polyribosomes on a message, a polyduplosome. A little more subtle is way back when the Earth rotated on its axis faster. And there were more days per year. It's hard to know exactly because when this is happening, it's a fairly rapid slowing, I think, but easily could be twice as many days per year. So all of this has the possibility that the generation time might correspond to a solar year, which would be nice. But even if it takes the 8 years, there's a window of RNA life of 200 million years or such. So that's 25 million generations, far more than is needed for a lot of serious evolution. So I want to talk about triplet decoding. So here's the original cartoon of doublet decoding. The concept of the step size and the elongation cycle is actually a little subtle. There are several concepts that have to be made congruent. So the original doublet decoding, the idea was you read a codon of 2 nucleotides, you add a duplicon of 2 nucleotides, you shift the dRNAs from one site in the duplosome to the next, and in that process, bring along the template by 2 nucleotides. But what's important is that the length that you add be exactly the length that you shift the template by. The reading length just has to be 2 or more. So if we look down here at protein translation today, we say it's triplet decoding. It reads the 3. It adds not nucleotides, but an amino acid. It shifts the tRNAs by site and moves the template, the message, the full codon. So the suggestion is that, well, we might be reading something like the modern ribosome 3, but add 2, shift the site, and move 2. And I want to pursue that just a little bit, just mainly because it's fun. Okay, so the genetic code and the idea of its redundancy. So with 20 canonical amino acids, it knew that if we had a comma-free code, we'd need at least codons with 3 nucleotides, making 64 codons, 64 triplets. But then it was discovered that there are not nearly so many anticodons. You don't need 64 different tRNAs to deal with that. One explanation was the position 34, the wobble nucleobase. Could read more than one thing on the message. A little later on in the chloroplast, discovered the mitochondria that super wobbling, that one nucleobase can read all 4. So I want to talk about frame and frame shifts. So the most common frame shift is a -1, and 2 usual pictures of this is that it's either the slippage of a single tRNA on the message, or the slippage, synchronous slippage of 2 adjacent tRNAs on the message. And I want to introduce the possibility of a third situation, a double scrunch. So just show this. So the usual view is kind of associates frame shift with translocation, not with decoding. So in the post-translocation complex, before it's loaded at the A site, the peptidyl tRNA might slip -1, and it's favored by an RNA sequence, a slippery sequence. It's also favored if we deplete the pool of charged tRNAs for that A site. So if it sits not filled for a while, this slips more likely. After peptidyl transfer in the pre-translocation complex, it's thought that the deacyl tRNA and the peptidyl tRNA now could together slip by a nucleotide. And this was favored by certain sequences. And then in viruses and bacteria, different things, there are places where they program such a slippage to happen by having a slippery sequence and downstream at the right distance, a hairpin or pseudonaut or something to encourage it. So this is the top. This is the 1 tRNA slippage. Here we were in the P site, and here we slipped. Here's the 2 tRNA slippage. Here we are in the P site, and we've loaded the A, and then together the module slips. This is scrunch. The thought is we've loaded the P site, and it's on the loading of the A site, a mistake is made that the peptidyl tRNA yields, doesn't defend the wobble position, and allows the new one to come in and read the -1 codon. And one thing I want to say right off this, whichever mechanism, they all have the exact same consequence for the protein made. So what determines the codon specificity and the frame, or the defense of the frame? The nucleoside modifications in the tRNA, particularly in the anticodon stem and loop. So the most heavily modified positions on tRNAs are the wobble base 34 and 37 just after the anticodon. The modifications on the wobble base can expand or restrict decoding. The other modifications involved with defending the reading frame, avoiding frame shift. So here's the idea that tRNAs normally stand apart. So the preserver reading frame, both in decoding and translocation, it's nice that the peptidyl tRNA, I guess I don't have a T there, it's an energetic minimum in the 0 frame. And then it must defend the wobble pair as the A site is loaded. So the A site decodes in the 0 frame. And then they both have to shift quickly and synchronously. So all the things that have been implicated in this include modifications on 34 that seem to be working at the P site. Something recently described as the wobble seal, which is a highly modified residue in the key position on the small subunit ribosomal RNA. This modification in eukaryotes and the matching one modified in bacteria. The modifications on 37 in the A site involved in cross-strand stacking, modifications of 34. And then in the translocation GTPase, the doorstop, the domain for tip residues, and sometimes in eukaryotes modification there, the dipthamide. Okay, so that's a lot of stuff involved in the correct framing and preserving the frame. And so I throw this out here. Can you have a frame shift without slippage or scrunch? So we want the peptidyl tRNA to yield the wobble position, and we want the aminoacyl tRNA to come in and read the -1, and then the 2 to shift synchronously. This might be favored again by under modification of any of these RNAs or a lack of the GTPase. So the reason I went through this is to suggest that maybe this was the normal mode for the dRNAs on the duplication. They may normally have scrunched together in a 2+3 module. So the idea is triplet decoding but doublet addition, so that the duplicate RNA yields the wobble pair normally. The duplicon dRNA decodes the -1 frame. They shift synchronously. Of course, the RNAs are not modified in this RNA world because the modifications are all protein enzymes. So there's no modification on the tRNA or the wobble seal. Position 37, we suggest, doesn't exist. 36 is the end of the dRNA, and there's no translational GTPase. And so why would you do this? Why would you read more than you add? Well, you get a little bit longer mini helix, so you may need that additional stability for good fidelity. A possibility that you need it that long to make a toehold to open the dRNA. And a possibility it helps in monitoring that you've hit the end of the template termination. I'm not going to talk about the speculation. It's basically that the intrinsic movement doesn't have to be an integral number of nucleotides, only that afterwards you have to get it to 2 or 3, depending on your goal. So here's the notion of the toehold opening. So this is the A site charged dRNA. It's come in and sniffing out the codon, pairing with it. And to do that, it breaks its pairing with the duplicon. So this is the beginning of prying this open, which we say warming of the day was going to favor it, but kinetically getting that toehold start, that may be the difference between a cognate and a non-cognate dRNA entering the A site. There it is, accommodated and ready to transfer. And I'm agnostic about just how much is melded out and how much is not. Okay, so that's a cartoon to have a cartoon. So this is the slight adjustment where we're adding a wobble position to the anticodon. And then this is the modification of triplet decoding with doublet addition, the scrunch idea. Okay. I want to talk about what happens when you hit the end of the template and certain other things, but termination. So the polypeptide, the nascent polypeptide, is being built up at the PTC and then gets to the surface of the ribosome through the exit tunnel. The walls of the tunnel are mainly RNA. There's some protein loops that also line it. The tunnel dimensions about 100 angstroms long and 10 to 20 angstroms wide. So its diameter and length are enough to support a random coil of 30 or 40 residues, or a rather longer alpha helix. Very near the outside, there's a slight widening called the vestibule, and this is kind of the fate of the niacin polypeptide. So it starts folding inside the tunnel, alpha helixy. It doesn't have to keep that. It's entropically encouraged to be an alpha helix in the tunnel, but inside the vestibule, people have shown that it can form a beta hairpin or even an entire zinc finger module, a very tiny fold. Anything bigger, any bigger folds have to occur outside the large subunit. If the protein is destined to cross a membrane, they go outside, or some membranous compartment has to be secreted. That's coordinated with translation. This was the system that was set up by the time of Luca. So 30% of the proteome is transmembrane or secreted. So a hydrophobic signal sequence or a hydrophobic transmembrane domain is the trigger. And the signal recognition particle has got to recruit the ribosome, with the destined to be secreted protein to the translocase on the membrane. Protein termination. You have to release it from the tRNA in the P site and recycle the ribosome. What if that has to change if it's a duplisome making RNA? Well, the first thing happens that when you get to the end of the template, so here we've shown you may or may not be able to read quite to the end. So here we're able to read it, add the CU and the duplicon. We can't go any further. So we break these things apart. Here was the template we tried to copy. This is the duplicate we made, and it has still attached to it the final dRNA. So what's the polynucleotide exit going to look like? The walls of the tunnel are going to be solely RNA. This is pre-protein world. Maybe non-coded proteins are around, but anyways, electrostatic repulsion of the backbones, maybe hydrogen bonds. Maybe thinking about the message tunnel today, which is an RNA inside the ribosome, So here's another RNA now going. Folding. Perhaps inside the tunnel, small hairpins could fold, but anything bigger, any bigger secondary or tertiary folding has to be outside. Just for the record, how big would a folded dRNA be at the tail? So the 15 base pair extended stem, 8 nucleotide loop. So it's going to be more than about 50 angstroms long and about double helix diameter, so 20 angstroms diameter. So now we have the questions. We try to add the final duplicon. Exactly how far we go may depend a little bit on whether it's an even rod length template. We want to release the nascent RNA. It's possible the duplicate with the dRNA exits through the tunnel. And then the question, does it exit open or closed? Is the dRNA in the open or closed conformation? It's also possible we have to disassemble the duplisome to get the damn thing out. And then what about the end? Is there a maturation of the end? And is there a way to protect the ends of RNAs from the inevitable erosion from the fact that you can't really duplicate quite to the end. So here's the notion of the end erosion. Here's the template. This is as far as we could go with the duplicate, and maybe we cleave it. Maybe that's ribosome P. So this is what we free, and a charged dRNA left over maybe. So we see in this example, we lost the funnel 3. So I want to talk about recombination. Recombination is a big deal. Big deal in genetics, you know. The great things are to bring genes or RNAs from different lineages together in one community. And then if you could actually recombine them to make a covalent larger gene, especially interesting. So the duplosome may mediate recombination by forms of template swapping. There are 2 obvious ways this could happen. One, the duplosome disassembles, so it stalls halfway down a template, and it reassembles and resumes duplication, but now on a new template. The second one, it may be a swapping without a disassembly of the duplosome on an intact duplosome. I'm going to talk about that too, but let's first do this one. So it's pretty simple. Here's the duplosome, and it's stalled. We've separated the subunits. The subunit with the nascent chain reassembles, but on a new template. Here's the first dRNA extending it. So when we're done, we had 2 templates, the green and the orange, and the product we get is begins with the green and ends in the orange. So it's a recombination. So the tmRNA system for translation, so for translational surveillance and ribosome rescue. It sounds too good to be true the first time you hear of it. So these are small RNA that look a bit like a tRNA, and they look a bit like a messenger RNA. So the tRNA-like domain it starts at the 5', but it's also it ends at the 3', and it looks enough like a tRNA that it can be charged by an alanine aminoacyl tRNA synthetase. And then here's the messenger-like domain nested in some pseudoknots, but it's got an open reading frame. It has no start, but it has a stop. So the idea is that when the ribosome stalls at the end of a nonstop mRNA, this complex of the tmRNA and its protein companion loads into the A site. There's a transpeptidation, so this alanine picks up the nascent chain. There's a translocation, and amazingly, there's a template swap where this little piece of RNA gets into the message channel well enough that it can be translated through the open reading frame to the end. The open reading frame encodes, I don't know what, looks like 10 amino acids, plus the alanine here, make a tag, so the nascent chain can be recognized and targeted for degradation. The nonstop mRNA can be destroyed, and the stalled ribosome recycled. This is, I think, a cryo-EM paper showing, here's the complex, the small protein B complexed with the tmRNA, and the alpha helical tail of the protein here sniffs out that the message channel is empty and facilitates somehow this message-like domain, filling the other part, the downstream channel so that translation can continue. And now on this swapped message. So here's the idea for the duplication-mediated recombination, where we swap what we're dubbing a donor template RNA. So here is a donor template RNA. It looks like a dRNA. It has the duplicon and the D stem. Coming down to where the anticodon is, instead it has something else, any length you want. So that's the template-like domain. So here we hit the end of the template, and we've swapped in a donor template RNA, and then we've resumed on its template, producing this recombinant product. So I'm going to finish. We want to go now from an RNA life, what we called, I think, late RNA life, where RNA makes the RNA via the duplisome, to protein life, and then beyond. So this is the idea going back to Campbell, that RNA is made randomly, then made by copying, non-enzymatic, but now duplisome assisted. And this is not a world of coded proteins, but there may be random polypeptides available. Now we're getting to early protein life, so they're going to have a ribosome, a tricked ribosome doing protein translation. But we still have a late duplisome that's responsible for duplicating the RNA genome. And then eventually we're going to retire the duplisome and have a protein RNA-dependent RNA polymerase. So this late RNA life with the random proteome, fewer amino acids, presumably, than today. The ribozymes may assist the synthesis of amino acids. The activation shows that with ATP, reminiscent of the modern charging, and some mechanism of polymerization. It's not in any order, it reflects presumably just the concentrations of the pools of activated amino acids. So what do these short random peptides do? They have simple compositions. It's suggested that they work. They get selected by co-assembly. So it's not natural selection in that it's a heritable thing. It's co-assembly in the working cell of the moment, ribonucleoproteins, and maybe some proteinaceous membrane pores. To get the protein translation, we need to do something about our dRNA. So we're suggesting it's a hairpin and it's molten. And we need a tRNA that's a clover leaf, and at least for a long time was viewed as being a fairly rigid molecule. So I want to just race through this model. It's a single hairpin model for the origin of the tRNA. So the tRNA today corresponds, the 5' end of the tRNA is one ancestral dRNA. The double hairpin model, where two homologous DNAs are glued together to make a tRNA, and a polyhairpin model. So all these papers showed the cloverleaf, but a second double hairpin model, he said, well, it looks like that might be a feasible structure. So here the anticodon is connecting between the two hairpins, an extended D loop, but extended T. It was discovered that in vitro, under certain circumstances, that double hairpin exists. One evidence of it was discussed in this paper, taking some, not every tRNA, but some tRNAs from Drosophila, and discovered that you can see these arrows, that a portion of the population misfolds or refolds as a double hairpin. And that can be detected by adding an RNase P, bacterial RNase P, clipping it. Another suggestion, sequence gazing, is that the modern cloverleaf tRNA might have emerged out of two hairpins, identical hairpins with an extended stem that are now melded apart and repairing. And in this model, we end up with the anticodon at one end and the residues important in the charging specificity at the other. Genomics has revealed, not all tRNAs have an intron, but in all kingdoms of cellular life, there are tRNAs with an intron. The canonical position is between N37 and N38, so just a smidge after the anticodon. In bacteria, introns that are found at that position are not the ones processed by the protein endonuclease, but they're self-splicing group 1 introns. And there's a speculation that somehow the eukaryote archaeal processing to remove them is evolved as a protein-based way of removing what originally was a self-splicing group 1 intron. Another little aside is in some archae today, transplicing assembles the functional tRNAs, and here's a situation where 3 different 5' halves can assemble with the same 3' half to make 3 alternative tRNAs. So the polyhairpin model, it's just taking note that when we duplicate an arbitrary template, we end up with a donor RNA stuck at the end that we're not quite sure what to do with. If we duplicate a donor RNA, we have a donor RNA with a hanging donor RNA, and we do it again, donor, donor, donor. We keep, each time we build up every duplication round this longer polyribbon, polyhairpin. And the dRNAs are not the same, but they alternate. The codon duplicon relation anticodon duplicon relations alternates. So if we can cut this down, maybe by a ribosome P mechanism or so, we can get these kinds of products. Okay, so this is just a hint that maybe the notion that 2 dRNAs equal 1 tRNA is worth exploring. So early protein life, exploring the proteome. Okay, now we have coded proteins. Immediate problem, given an RNA, you have to decide, is this to be duplicated or is this to be translated? It's the promises of a huge muddle. We have to distinguish when we want to use it as a template and when we want to use it as an mRNA. It's possible that this would justify the evolution of cap or tail structures. We have to know, do we want to use dRNAs or tRNAs if we're doing RNA duplication or doing protein translation? So maybe those can be distinguished. Maybe that's the 5' phosphate, maybe the universal tail, maybe some of the base modifications. Just things to consider. The actual some itself, the duplosome versus the ribosome, how different are they and how do you not confuse them? So base modifications, maybe. It's possible that the 5S RNA is there as a vestige of what distinguished the ribosome from the ancestral duplosome. Okay. But anyways, huge problem. Not just choosing when to do which, but not to muddle it. Not to mix up things that maybe can't be mixed easily. Once we've decided to translate, there's a problem of where to start the translation and whether to export the polypeptide, where to stop. And then the whole question of the origins of our peculiar genetic code and all the theories about that. So we've got Protein Life off and running. We want to exploit this. So improvements in protein translation, so the modification of nucleotides, tRNA splicing, ribosomal proteins, the protein components of RNAsP, of course the amino acid tRNA synthetases. Only thing I'm going to mention here is the translational GTPases. So we've improved protein translation. We've got to improve RNA replication too, and apparently going over to a protein polymerase. And I won't talk about it, but clearly then you have to retire the duplosome. That may be a simple thing or not so. And then all the other good things you can do with proteins to improve your membrane compartments, metabolism, energy sources. So I just want to mention these two, and then we're done. Well, first of all, we need to improve the speed and fidelity as we go from duplication to translation. Substitution errors, misincorporation, they're not so different. For the RNA duplication, we might put in the wrong nucleotide, or perhaps two, the entire duplicon could be wrong. And protein translation, we put in the wrong amino acid. But frame shifts are entirely a different matter. And duplication of frame shift is local. So it shifts from the even to the odd reading frame. Change which loaded dRNAs we would be using, but the product is only going to be changed by an indel, one nucleotide indel. When there's a translational frame shift, it's catastrophic. So we shift from the 0 frame to -1 or +1. We get a protein that's just unrelated in sequence, and it's likely to terminate prematurely. So the requirements for frame preservation are low in the duplosome and high in the ribosome. So I'm talking about the translational GTPases. So in the ribosome, at a common site, swapping in and out different homologous GTPases improve the fidelity or directionality or both of steps. So this is the way it works with high fidelity today, and I guess the speed too. So let's just look here at the elongation cycle. So for reading, it's in bacteria, it's elongation factor 2. Peptidal transfer itself has a slight free energy advantage because the ester bond is less stable than the peptide bond that's formed. So this is the free energy available from the hydrolysis of GTP in the reading and the translocation and then the peptide transfer. So about 26 kcals per mole per step. If we come over here for the duplisome, just that argument about the prying open in the warm and clamping closed in the cold, we have a difference of about 12 kcal per mole, and the phosphoryl transfer is isoorganic. So we have about minus 12 net per elongation. And a little throwaway here about what we might get by folding the products. So the translational GTPases are great contributors to the speed and fidelity of protein translation. And the idea is this thermal motoring is all the duplisome had to go by. This is the final slide. So we want to add DNA. So in early DNA life, it's a minimum. We had to be able to make DNA from RNA. Says reverse transcriptase. And we had to be able to make RNA from DNA. So that's the universal transcriptase. It's suggested that then it's actually, this is LUCA maybe, it's later steps and different branches where the modern dependent DNA polymerases emerged. Okay, so this is a life that uses DNA, but DNA replication has to go through an RNA intermediate. And here is the central dogma: DNA making DNA, DNA making RNA, RNA making protein. Thank you. Tell me that crashed. I recorded it. There's a wrinkling.